You're able to see?
Yes, ma'am.
Yes, ma'am.
Yeah. So last class, I think we had learned about the internal anatomy of Pinus root and young stem and old stem of Pinus. That means secondary growth also you had studied. Now coming to the needle, that means leaf. Now in case of Pinus, you all know that foliage leaf or the photosynthetic leaf is referred to as needle because they are all sharp pointed needle like or acicular we call. So generally they are triangular in outline and they are sharp and needle-like, and the dwarf shoot along with the needle together, they are called as foliar spur. If you remember when I had explained the morphology, I had mentioned to you about this. And
Yes.
depending on the number of needles, it may be mono, uh, yeah, unifoliar spur, bifoliar spur, trifoliar spur,
Trifoliate.
yeah, like that it can vary. So now when you take a section of this particular needle, this is how it will, somebody is trying to join.
Yes, ma'am.
Yeah, ask them to join, no, quickly. 23.
They have joined, I believe.
Yeah, okay, fine. So okay, coming to the transfer section of pine needle, you can see that it reveals epidermis, cortex, and stele. This much you know. Now, when you take a section of pine needle, it's quite different from the other leaves. See, when you had seen with in case of cycas, it was quite different. And in case of Pinus, as I just told you, they are all needle-like leaves, just triangular in outline. They are very tiny. When you take a section, it's a very small, tiny section. And I hope you remember, you would have already seen the slide in the practical class and how beautiful the structure is, the internal structure of pine needle, especially. And earlier, like we used to make the students take section of pine needles and beautiful small triangular sections they would take, and they would subject them to double staining, fast green and saffronin, and beautiful staining. When it takes, it appears so nicely, the slides. Now coming to the internal structure, so you can just see that when you take a transfer section of the spine needle, it will as usual reveal epidermis, and instead of telling cortex, usually here we call it as the mesophyll region, and then followed by the stellar region or the vascular bundle region. Somebody else has come now.
I think so, ma'am.
Yeah, because I can hear the sound, no?
Yes, ma'am. She has joined now.
Okay,
Thank you.
fine. So epidermis, it is as usual, it is single layered, and then it is slightly made of thick walled cells and covered externally by a very thick cuticle. Because you know that this thick cuticle is one of the xerophytic feature, and it shows sunken stomata. Again, sunken stomata also is a xerophytic feature. So epidermis is not continuous, it is discontinuous due to the presence of sunken stomata. So this is what you will be seeing here. And then the hypodermal region, that is the region just below the epidermis, is made up of what is it made up of? Hypodermis is made up of sclerenchyma cells. Again, sclerenchyma cells also, presence of hypodermis with sclerenchyma cells also exhibits a xerophytic feature. So it in fact provides mechanical strength or strengthens the leaf tissue. And then you have the mesophyll region. Now, the inner region. So, mesophyll region means it contains cells with chloroplast or chlorophyll bearing cells are there. So, these cells in case of Pinus are quite different. They are specialized, and these are parenchyma cells with peg like projections or invaginations to the interior of the cell containing chloroplasts. Now, these specialized cells are called as armed parenchyma, ARMED. Okay, so they are called armed parenchyma. So, this is what you're seeing here. If you observe, see, you're able to see the section. So,
Yes,
as
ma'am.
usual, you can see the opening stoma. So, this is the stomata. So, outermost epidermis, externally covered by thick cuticle, interrupted by sunken stomata. Then hypodermis is sclerenchymatous, and internally the mesophyll is armed. That is, it is called as armed parenchyma. And then the vascular bundles, if you just come to the vascular bundle region, they are said to be collateral and closed type of vascular bundles. Now, what are these collateral bundles? That means you can just see both xylem and phloem will be present in the same radii. So, you can see that it is usually surrounded by endodermis and pericycle.
Pericycle.
Yeah. And one more thing you should observe, even in the mesophyll region or the hypodermal region, or you can just say cortex region, you will be observing the presence of resin ducts leading to resin canals. That I just forgot to tell you, because this Pinus pine trees are rich in a resin, namely turpentine. So, one more thing, what you should know is here the endodermis also will be covering the vascular bundle region, followed by what is called as pericycle. Now, in case of pericycle, generally it shows different types of cells, parenchyma cells, and then tracheidal cells, and some of the parenchyma cells are found at the base. Okay, so the whole it becomes almost, see, it has albuminous and tracheidal cells, we say. So some of the parenchyma cells, they are rich in proteins and starch. So such cells are generally referred to as albuminous cells. And there are also tracheidal cells. And these albuminous cells, they are generally near the phloem. See, this is the phloem, what you're seeing here. Now, exactly at the center of the vascular bundle, there is a T-shaped girdle or patch, T-shaped sclerenchymatous patch, which is also called as girdle. Okay, so this is the sclerenchymatous patch which you are seeing, which separates the vascular bundle into two. See, two vascular bundles. So about just below the T, arms of the T, you have the phloem, and lying next to the phloem, you will see these albuminous cells. Albuminous cells, you can see small parenchyma cells with small dots. It is shown. I have just shown very few cells. So these are the albuminous cells. And there will be certain other cells which are referred to as the tracheidal cells below. Okay, these are all xylem elements. Below the phloem, you have xylem, and then you have tracheidal cells. Now, these albuminous cells, tracheidal cells, and the parenchyma together, they form what is called as transfusion tissue. Just like the previous, what is that? Form what we studied here also, there is no lateral vein, only the midrib region you will find. So the lateral conduction will be by these transfusion tissues. It helps in the movement of the nutrients also. So these are called as transfusion tissue. So this is about the what do you say, structure of the internal structure of the leaf. Now, pericycle, as I told you, it also has a T-shaped sclerenchymatous patch. So all these are found. I mean, this presence of transfusion tissue itself is one of the important feature in case of pine needle. So this sclerenchymatous patch, separates the vascular bundle into two. So generally they are conjoined collateral vascular bundles, but they are closed type of vascular bundles. Then what else I had to tell you? Thick cuticle, sunken stomata, sclerenchymatous hypodermis, transfusion tissue, and girdle-shaped or T-shaped patch of sclerenchyma. All these are adding up to the xerophytic characters of pine needle. Have you understood?
Yes, ma'am.
Yes, ma'am.
Oh, wait. Once again, if you can just see. Okay. Same thing. I think this is madam's diagram and this is from my record. Okay. Yeah. Next. The same thing. What you're seeing here, tiers of root, stem, leaf, everything is put together. Once you can just brush through, go through. I have explained everything. Just one look. Just have a look at all the three. Last class, just recollect. That's all. Nothing else. Over. One more thing, for your internals, whatever portions is completed, you will have till there. That's what I think madam has told you.
Yes, ma'am.
Yeah, because you will be having MCQs, no? So the portions will be very less in the sense for us to give you questions, options, no? So that was one thing. So shall I continue?
Yes, ma'am.
Yes, ma'am.
Next, coming to the reproduction part. So now, in case of Pinus, so plant body is said to be monoecious. That means both the male and the female reproductive structures will be found on the
Same.
plant, but
Same
the
plant.
cone, yeah, same plant, but the cones are separate. Male cone and female cones will be separate. Okay, so unlike the previous form cycas, in cycas what happened, they were dioecious. Male and female plants were separate, whereas in case of Pinus, they are said to be monoecious. That means both the reproductive structures are found on the same plant. Only thing is the cones are different. And one more thing, what you should know here is male cones will be generally smaller in size compared to the female cones. Female cones are larger, male cones are smaller in size, more in number, and they are slightly soft in nature, unlike the hard and woody female cones. Okay. So you should remember, generally these cones will be borne in the axil of the dwarf shoots. So next slide I will just go to. So see, in the first diagram you can see a cluster of male cones. Okay, so dwarf shoot you can see. So axil of the scale leaves, generally these male cones are borne. So there are clusters of male cones like this in the terminal region of the stems. So you can see one picture here also, cluster of male cones. Okay, and generally, as you know, even Pinus is heterosporous. That is why it will produce two different types of spores. What are the two different types of spores? Microspores and megaspores. Yeah, microspores are nothing but it refers to the male reproductive structure. Female reproductive structure refers to the megaspores. So generally, if you take one microsporangia, I mean, if you just take, not microsporangia, if you just observe the structures of Microsporophyll. Now, generally, what are these cones made up of? The cones are usually made up of, what is a cone? Male cone. It's an aggregation of
Microsporophylls, ma'am.
Sporophyll.
Yes, very good. It's an aggregation of numerous sporophylls, microsporophylls. So they will have generally, they are just few centimeters in length, male cones, just around 2 or 3 or 4 centimeters maximum. Okay, and they will contain usually 50 to 130 or 135 microsporophylls in one male cone. Okay, and diameter also, they will be not very thick. Generally they are just less than a centimeter in, less than a centimeter in diameter. So they will have a central axis around which you will find numerous microsporophylls arranged in the male cone. Now, when you take one single microsporophyll, see, this is what you are seeing in the second picture here. So a single microsporophyll showing a stalk, a sterile stalk, by means of which it is attached to the axis, central axis. And then the microsporophyll, it as usual extends into a sterile tip. What is the tip called? Hmm. Sterile tip. Yes, apophysis. Now, just below the apophysis, above the stalk, you will find a pair of microsporangia. So generally microsporangia are in pairs. Okay, and then the microsporangia are nothing but the pollen sacs. Now these microsporangia or pollen sacs, if you just observe in this diagram, you can see there is a central line of dehiscence. Okay, now these microsporangia will bear numerous
Microspores, ma'am.
microspores or pollen grains. Now one of the unique or feature of Pinus, the pollen grains are namely they have
Wings.
yeah, very good. They have winged pollen grains. I think in the last class I had just mentioned to you.
Yes,
We had just discussed
Yes,
about this also. Yeah.
ma'am.
So each of the microspore or the pollen grain, if you just see, it shows the presence of a pair of wings. Okay, a pair of wings are present. And then if you just observe, see, it shows what are the wall layers of the pollen grains called? It can be differentiated into
China
Final
textile.
intine.
exactly outer exine and inner
Intine.
intine. Now here what happens, in between the two there is one more layer. In case of Pinus pollen grain, there is another layer which is called as exointine, which is the middle layer. Okay, exointine is the middle layer which usually extends on either sides in the form of wings, which helps in dispersal of seeds, dispersal of the spores also, you can say. Okay, so, and of course it encloses the nucleus and the cytoplasm. So this is about the structure of the winged pollen grain of Pinus. So it has three layered wall: outer exine, inner intine, and then the middle
Exointine.
exointine, yeah, which is extending on either sides in the form of a pair of wings. And then the pollen grain encloses nucleus and the cytoplasm. So, like some of the scientists, they have just told the exine only probably is thick, and it is not the exine, but another layer is there. Some of them opine that it is just two layer and exine itself extends. But some of the scientists, they said exine is the outer cutinized thick layer, intine is a smooth layer inside. In between, there is a third layer, or you can say a middle layer, which extends to form the wings. So it is not from the exine or the intine the wings are appearing, but it is another layer in between the two, which is called as exointine, which extends to form the wings. So that's what, there are different opinions. Some of them say it is only made up of two layers, whereas some of them say in between the two, outer and inner layer, there is another layer which will produce the wings on both the lateral sides. So of course, the pollen grains, as you know, they are all just uninucleate. They will have a single nucleus with little cytoplasm with reserve food material, and all those things will be normal. So then what else I had to tell you? I think this much you should know for the male reproductive structure. Next will be the female reproductive structure. So female reproductive structure, they are also found in the terminal regions of the stem in the axil of the scale leaf. Yeah. See, this is the male gametophyte, microsporogenesis, and yeah, formation of the spores, pollen grains. This is the longitudinal section of the male cone. These are all the sporophylls. This is central axis, and this is a single microsporophyll. These are the two microsporangia, central line of dehiscence. This is the sterile tip apophysis, the stalk, which is also sterile. This is the winged pollen grain, exine, intine, and the exointine extending in the form of lateral wings. Okay.
Yes, ma'am.
Yeah. Only you should know the structures. Yeah. Same thing again. One more set of diagrams. See, lot of microspores or pollen grains are seen within the microsporangia. Yeah. Now coming to the female reproductive structures. So even here in case of Pinus, you know that there is formation of the female cone. See, you can see large female. Have you seen pine trees, pine cones? Any of you?
Ma'am, in lab we had seen.
Yeah, lab you have seen. From the tree I'm asking. What picture is seen here, no? No.
No, ma'am.
No.
No, ma'am.
Okay. Okay. Some like right now it's okay. In future, maybe with your family or some, of course, right now leave the situation. If you happen to visit any hill station and you observe these cones, they would have fallen on the way itself, where a lot of pine trees will be there. No, you will know, okay, these are pine trees, and so many of the pine cones have fallen. So then, when you go, hill stations, you can observe these cones. Beautiful they are, large and hard and woody they are. So now we will just look at the structure of the female cone. So female cone also is made up of numerous leaf-like structures which are called as megasporophylls. You know that. So female cone is a compact cone-shaped structure which has a central axis, okay, surrounded by numerous megasporophylls. Now these megasporophylls, which are spirally arranged around the cone, has two structures. Do you remember the two structures?
Yes, ma'am.
What are the two structures? The bract scale and the ovuliferous scale.
Yes, ma'am.
Yeah. So the bract scale is the sterile scale. The ovuliferous or the carpillary scale is the fertile scale, which will bear the ovule. Okay. So this much have you understood now?
Yes.
Yeah. So that means each of the megasporophyll has two parts, bract and the ovuliferous scale. Bract scale and the ovuliferous scale. Now, this ovuliferous scale is also called as carpellary scale because pistil or carpel is the reproductive structure. So it bears the megasporangia or the ovule, and then it bears the egg or the megaspore. So this much you should know. So now these are the ovules which are seen here. See, all these are ovules. So now you are finding the ovules on the ovuliferous scale. So each of the megasporophyll has a single ovule or the megasporangia, and then it has also the megasporophyll also has another appendage which is called as the bract scale, which is the sterile scale. So this you have understood. Now one more thing you should remember. Somebody is trying to join now. What is the time now?
2 o'clock.
2 o'clock, ma'am.
So these cones, if you just observe, see, this is the young cone, and this is slightly a larger cone than this. So this says first year, and this is the second year, it says. Okay, if you just observe all these cones will, the megasporophylls, if you observe, they will have a small rhomboidal projection on the surface, and these projections are called as umbo. Do you remember?
Yes.
Yes, ma'am.
Yes,
In
ma'am.
practical class, you
Yes,
might
ma'am.
come across this. Yeah. So, yeah,
Yes,
we will
ma'am.
just look into the structure. You're able to see this now, whatever I explained.
Yes, ma'am.
Yes, ma'am.
No, previous slide. I don't know why I have given some effect. Okay. Now, here if you just observe a single megasporophyll on the left side, if you're just observing, again here also it has a sterile tip which is called as
Apophysis.
very good apophysis. It has a stalk at the base, and on either sides of the what do you say? On either sides of the Bedian scale or this portion, it will show the presence of a pair of ovules. See, these are the two ovules or the megasporangia. So this is what you're observing here. So these are called as the Oviliferous scales, if you remember. Now, so this is one such megasporophyll. So this is the megasporophyll. Sterile tip is called apophysis, the stalk which is also sterile, and then these are the two ovules or the megasporangia. Yeah.
Ma'am,
Yeah.
ma'am, will each ovuliferous scale have single ovule, ma'am?
Yeah.
Yes, ma'am.
That means one megasporophyll will have two ovules on either sides. That's what.
Yes, ma'am.
So
Thank you.
this is one ovuliferous scale and this is one scale. It's like that on either side. Okay.
Yes, ma'am.
Two.
Yes, ma'am.
So now what you're seeing in the center is the LS of mature ovule. So we need to study the structure of the mature ovule. So we will just look into this in the next slide. Okay. Do you remember Pinus ovule slide, which was focused
Yes,
under
ma'am.
probably dissection microscope? Did you observe it under dissection microscope?
Yes, ma'am.
Yes, ma'am.
How were they? Do you remember? How many ovules were there? Don't remember.
Ma'am, the color of it I remembered little bit, ma'am. The slide color.
Huh.
Yes, ma'am. The color was the same as, yeah.
Yes,
What you're seeing here, no? But
Yes,
you
ma'am.
don't remember what is that? How many
the
ovules?
details.
Twin ovules, no? That is what I wanted to know. Twin ovules were not focused. Pinus ovule, twin ovules are there actually. You have not observed. You've forgotten. Was it not mentioned in the class, practical class?
Yes, ma'am, it was there.
You
Ma'am,
remember now?
it was a round structure, ma'am, and in between we had a small line, ma'am.
Yeah, but actually Pinus, it is actually twin ovules. When you see under the dissection microscope, you can see twin ovules. That is how you
Ma'am, it was
Hmm.
pink in color.
pink in color.
Yes, ma'am.
Yeah, ತೋರಿಸಿದಾರ.
For our lab, the color was the same, ma'am. In what you have
Yeah,
recommended.
that's why I'm telling. Because this is the one which was supposed to be shown twin ovules with green, no? Green color stain. Maybe they have shown you some other slide then. Generally, for Pinus, we show the one with the twin ovules. Okay, anyway, now we will just look into the structure of the Pinus ovule. As usual, when you take a longitudinal section of the ovule, it has the wall. The wall is called as, what is it called? Integument. Yeah. Now here also, in case of Pinus also, it opens by means of an opening called micropyle. Okay, the opposite end is the chalazal end. The micropyle will lead into the micropylar canal. Now the wall or the integument is three-layered. What are the three layers? Outer and inner fleshy sarcotesta, and the middle layer is hard and stony, yeah, which is called as sclerotest.
Sclerotest.
So these are the three layers of the integuments. Now, if you just observe at the center, it has the nutritive tissue, which is called as
Nucellus.
nucellus, and the micropyle will lead into the micropylar canal, okay, which is not very, I mean, not micropylar canal. It will lead into the pollen chamber. Now here, in case of Pinus, pollen chamber is not very clear. Pollen chamber is not very clear, and as usual, the ovule will have a stalk which is called as, of course, the funicle, and the base of the ovule is called chalaza. This much the inner wall of the integuments, it will be fused on all the sides except near the micropylar region with the nutritive tissue, which is called as nucellus. This much Now, in the center, what does it contain? At the center, what is present? That's all I want to know.
Nucleus.
Gametophyte.
Female gametophyte or the embryo sac. So if you just observe now, nucellus has formed slight projection in the form of a beak, nucellar beak, which leads into the archegonial chamber. Now this archegonial chamber is present towards the micropylar region. So towards the micropylar region, you will find the archegonial chamber, which will show the presence of 2 to 4 archegonia. So archegonia, they are all very simple structures with probably 2 or 4 neck cells and 1 venter-canal cell and 1 egg cell. So the neck cells or cover cells, some scientists opine that it is neck cell. Some of them say it is the cover cell. But you cannot differentiate here into an elongated neck and a bulbous venter. Only thing is, you will see a small structure, a simple structure with a single egg cell, which is the megaspore, and then which contains 1 venter-canal cell and 2 or, as I said, probably 2 to 4 neck cells or neck canal cells, you can say, or they are also cover cells. So generally during reproduction, what happens? The pollen grains are shed near the pollen chamber, which is not very distinct. It reaches the archegonial chamber, okay, by putting forth the pollen tube, and then the male gametes or the sperms are released into the archegonial chamber. By then, the neck cells or the cover cells and the venter-canal cell will form a slimy layer. It degenerates and forms a slimy layer. They disintegrate or degenerate, forming slime, and then which leads to the easy movement or passage of the male gamete into the archegonia. So in the archegonium, the male gamete fuses with the female gamete to form the diploid zygote. Okay, so diploid zygote will generally later develop into the diploid sporophyte. Yeah, you can say it develops into a seed, and then finally the seed, when it is sown, it develops into a plant. Now, sometimes, many embryos are formed after the zygote is formed. In case of Pinus also, like some of the higher plants, in one seed itself, more than one embryo can be formed. And if in such cases, if it has more embryos in the same seed, single seed, then, generally it is referred to as polyembryony, formation of many embryos within a seed. It is referred to as polyembryony. So this polyembryony is also seen in case of Pinus. Same thing. Of course, as usual, you can see a definite alternation of generation in Pinus also between sporophytic phase and gametophytic phase. Sporophytic phase is large and it is dominant, whereas gametophytic phase is not a separate phase where it can be seen as an individual plant. No, here it will be just your pollen grains and the embryo sac. Those are generally referred to as the gametophytic generation or gametophytic phase. That I think you know by now, no? Because cycles also we have studied, and even in Pinus, it is the same thing. Yeah. Coming to the economic importance of Pinus, these are few uses you can just say. As you know, it is extensively used for manufacture of furnitures, okay, and also in construction, of course, and even lot of musical instruments, and and of course your turpentine is obtained from the pine trees, which has lot of applications, again in furniture industry. And then also it produces, since it has some antimicrobial properties, the resin, so it may also be used in production of certain disinfectants, pine oil. All these are very important disinfectant agents. And then they also have applications in pharmaceutical industries. Okay, these are few applications or economic importance, I can say. So with this we complete.
Pinus.
Yeah, Pinus. So I was thinking whether I should start with Needham, but I think it will become too much for you. What do you say? Same day, so much. I don't know. I'm feeling like that. So probably if we again meet, if at all, if you people feel like, then maybe we can think of
This Saturday,
hmm,
mm, uh,
Saturday. What,
this
ma'am?
Saturday, this Saturday, we since we are finished the classes at 1, mom. So
One. Okay. You just put me one message on Saturday morning. Okay.
yes,
Let me
mom.
see. Because I also have some classes. I don't know. I think it's at 1:30 or what. I don't remember now. So let me just see and then I will let you know. Okay. Because
Yes,
Saturday I don't have in the morning. I have little late only. One theory class of final year, I think. So let me see.
yes,
But you just keep me informed.
yes, mom.
Yeah. Then how
Mom,
many?
is it for only our combination, or should I inform